everybody. My name's Joanna and welcome to Pint of Science live tours, where we get scientists to take us on a tour of their lab. These scientists have pre-recorded their lab so we don't disturb the science and can see as much as possible. Another really special thing about today's stream is that it is the last of the Pint AU21 festival. So don't forget that if you missed out or would like to revisit anything in the festival, you can do so by going to our channel. Maybe this is a good time to also like and subscribe to the YouTube channel, as you will be able to see all of the fun stuff that's happening and see the future stuff that we might have in the pipelines. Now, if you haven't already, if you've enjoyed what's happening and you would like to support us, there is the opportunity to also help out by donating or maybe even buying some of our merch. If you want to donate, here's a quick little QR code. Oops, wrong side, my bad. If you want. Otherwise, my lovely volunteers who are helping us in the background will put it up in the chat room for you. And if instead you were looking to do some merch shopping instead, we have some really lovely merch that we would love to get out the door and into your homes. But for now, throughout the live stream, you'll notice that off to the right, you'll be able to have your own conversations or ask your questions. As this is meant to be an interactive tour, we would love to hear any of the questions you have that might be during the tour as we go through, or it might come to you after the tour. Just chuck that all into the YouTube live chat that's off to your right. Or alternatively, if you're more of a Twitter user, you can also use Twitter. Just make sure you put in the hashtag PintAU21. And that should also be in, let me get this right here. Oh, there we go. Yeah. I love doing things back to front. Now, you might also notice that during this stream, I'm going to be looking off in different directions. That's because I'm just interacting with my lovely volunteers who are helping me make sure that this all happens nicely and smoothly. So in the background, I have Tom who's helping me with the videos, and we also have Karen and Steph who will be checking all of your comments and passing them on to me. So thank you to them, lovely individuals. Now, when we do hold live events in pubs around Australia, we ensure that we recognise the local Aboriginal and Torres Strait Islander nations, one of many that makes up the continent that we now call Australia. Now that we're holding these events online and our speakers and much of our audience is streaming from around Australia, we want to acknowledge all traditional custodians of country, the first innovators and scientists of this land. We pay our respects to our Aboriginal and Torres Strait Islanders, elders past and present, and recognise their continuing relationship with the land, waters and culture. We especially would like to pay our respects to the Turrbal and Jagero people, the traditional custodians of the land on which the University of Queensland is built on, which is where we're going on our tour today, and also the Ngunnawal people, which is where I'm streaming from today, and also doing the video editing with Tom. Now, it's time to get on to our tour for today. Today, we are going to be looking at the Fertility and Development Lab at University of Queensland. And I have two special guests with me today. Instead of our usual one, we've got two lovely individuals. We have Dr. Emmanuel Pelosi and Yinan Ye, who are on screen. So welcome to both of you.
Hi, Joanna. Thank you. It's a pleasure to be here.
Thank you. Really very excited.
Thank you both for joining us. We really appreciate your time in showing us on this lovely, lovely tour. Now, before we get on to have a look at all your facilities, I'd love to hear a little bit about you both. So maybe what you do and where you work. Let's start off with Pelo.
Hi, so I am a group leader at the UQ University of Queensland Centre for Clinical Research, or UQCCR, as you will hear throughout this conversation. My research focuses on the genetics of reproductive conditions. So our work involves modeling some conditions that affect the reproductive system. And we do this by modifying the genome of laboratory animals. In our case, these are mice. And basically, we see what happens. So in a nutshell, we modify the DNA of these mice, and we look at what these modifications do during the development.
Awesome. Thank you so much, Pelo, sorry. And Yinan.
Hi, lovely Pint of Science audience. So I am Yunan. I'm a PhD student from Pello's neighboring lab, led by Professor Hayden Homer. So I am very lucky enough to be representing the lab, and I think lucky enough to be studying some of the most unique cells in our body. Biased opinion, but I think oocytes or eggs, sperm and embryos are really cool. So hopefully we can show you some of that today. And I'll just say how I came about in the lab was also through a tour that was before COVID. So it was nice to see everything. But hopefully today we can do the same. Thanks.
So are you saying that today your lab is looking for a few more lab members? Is that what you're
potentially. You never know.
All right, viewers, keep that in mind. It looks like Yunan and maybe even Pele are looking for some new lab members. So if you like today's lab tour and it's into what you need to do or want to get into, we'll flaunt that at the end, I think. Yeah, we'll give them some details of where to go. So I would like to head on to the tour, as we do have 2 facilities to look at today. But before we just head off onto the tour, just a reminder to everyone else that as we go through, if you have something of a question, want us to go back and look at something, just remember to put into the live chat your questions or go to the hashtag PintAU21. All right, Tom, can we have a look at the video? Let's get started. So, okay, we're in Queensland, but where in Queensland are we, team?
Yeah, I don't know if you put it up. I cannot see the video though. I'm not sure if that's on.
The video?
I cannot see it. I can see you, but I can't see anything else.
Well,
oh, here we go. All right, awesome. Okay.
Awesome.
So we are in Brisbane, and we are located in the RBWH campus. Excuse me. So the Royal Brisbane and Women's Hospital campus. So we're right next to the RBWH hospital, and this is the entrance of our building, UQCCR. And as we get in, you can see that there is an outpatient facility. We have a clinical floor on level 3, and these are the 4 main themes within CCR. So we have cancer, fertility, neuroscience, and infectious disease. And this is the entrance of our lab. You can see that it's quite secure. You can't get anywhere without a badge. And I would lie if I said that I never forget it. But so this is the actual lab. So this is the office space, a lot of offices and desks where our students and staff sits. And this is a desk of actually my postdoc, Alex. And we can see on the screen here, on the left, there is an immunofluorescent image. So we actually, this is called mesonephros. This is an embryonic structure that will develop later on in the reproductive tract, either the male or the female reproductive tract. And those cells that you see there, with colored in pink or magenta and green, those are cells of the female reproductive tract. The Mullerian duct is called. Those are the pinkish one, and the green ones are those that it will potentially develop into the male reproductive tract. On the right, that's a vector. It's a circular molecule of DNA. And this is what we use in the lab a lot. In any molecular biology lab, we manipulate vectors and plasmid to add or delete genomic sequences. Then, depending on what kind of experiment we're after, we use these vectors to basically modify the genome of laboratory animals, for example, or just cells.
So, okay, I might have missed it, but what animal, is it an animal or is it just a cell that we're looking at?
So that's actually a tissue from a mouse. So I believe that this is a tissue from a mouse that is 13.5 days after fertilization. And so this structure here that you see in blue is actually a piece of tissue that we took from a developing embryo. And then we applied some fluorescent antibodies to visualize specific cell types. And the cell types that make the epithelium of the called Mullerian duct, which is the duct that will develop into the uterus and the vagina, those are the magenta ones, whereas the green ones are those are epithelial cells, but they will develop into the male reproductive tract. So epididymis, vas deferens, and so on. So one thing that that you can actually appreciate here is that, and maybe not everybody knows, during our embryonic development, both male and females, we have both structures that can develop into both the male and the female reproductive tracts. Then in females, the Wolffian duct will regress, whereas the Mullerian duct will continue developing. And the opposite occurs in males, where the Mullerian duct regresses and the Wolffian duct will continue on and develop into the full male reproductive tract.
Wow, there is a lot going on. Now, so, okay, I have a lot of questions going through my head right now. I'm trying to just get one out at a time. So
go for it.
When it comes to having to stain these cells, is this the true colors that we would see? Like, so when you go to look at it straight onto the microscope, will you see this exactly like this? Or has there been some behind the scenes magic to be able to look at it so pretty like this?
This particular experiment was a lucky one. This was exactly what happened. So no editing was required. But usually, yeah, there is some editing that that you can do. For example, every color that you use has a separate wavelength, and so you can basically in post-processing you can mix up the color. So you can choose to represent the green. The cells that you see green here with a different color, for example. But pretty much this is what the real image is. So you have fluorescent dyes, let's call it. They're actually fluorescent antibodies that bind to specific proteins within cells. And these fluorophores, so these fluorescent antibodies, they have particular colors. So they come out in the green channel or in the red channel, and so on. But so aside from just mixing up colors, yeah, I would say that this is pretty much, like a very straightforward representation of what really looks like an experiment, well done.
So this gets Pillow's tick of approval, I see.
That's right.
Well, alrighty, let's keep going with the tour and see what else we can find.
So this is in this. Oh, this is actually the looks like a sequence of that vector that we see on the other screen. So here you can actually see the actual sequence, the DNA with the 4 nucleotides, A, T, G, C, the 4 letters of our DNA represented in a sequence.
Ooh. Whoa.
And this is the classic, it's called chromatogram. This is when you do sequence a fragment, a piece of DNA, this is what comes out of the sequencing machine. So each peak that you see there, that's a nucleotide the machine has detected. These are color-coded, and the T's are in red, the A's are green, C's are blue, and G are black. But so this is the actual sequence of DNA that was put into the DNA was put into a machine to be read base by base, so letter by letter.
Right, and this DNA sequence that we're looking at, is this from a person, a mouse, or like have you just stolen blood from your honors students or something?
We don't go that far, just not yet. This is just a mouse. This is a mouse sample.
Oh, okay. And just out of curiosity as well, how different is the mouse DNA compared to like using human DNA? Why can we use mouse DNA rather than just using the honors students' DNA?
Oh, because the DNA of the mouse is basically pretty much exactly the composition is exactly the same as a human. However, there could be some genetic differences. And so when we want to do some genome editing, of course, we don't do this in human, we do this in the mouse. This particular fragment of the DNA, well, I don't know exactly this one in particular, but in general, what we do in the lab, we take a gene and we put it into a mouse, for example, to see what happens when we do that. Or we could do the opposite. We could synthesize in the lab a particular piece of DNA that has some modification here and there, and substitute the gene in the mouse genome with our construct that we did synthesize in the lab to see what that variation does. And so all these manipulations, of course, are done in cells or in animals. But if we were a lab that does sequencing of human samples, so for example, to detect a variation in a human genome, the sequence would look exactly like this. And we would use, of course, a DNA from a human, from a real person.
Now, you mentioned changing, as in like putting in synthetic sequences and all of this talk. I've heard only the word DNA. Now, does this also apply to RNA, which is the single-stranded version of DNA? Would you be able to do a similar thing of cutting things in and putting it into RNA, or is that unheard of? Have we just discovered something new here?
No. So what we do, we actually, so I'm going to talk a little bit later about, you may have heard the technology CRISPR, which is a genome editing techniques that allows to modify the genome of a cell, for example. And that particular CRISPR genome editing technology uses RNA sequences, so RNA fragments, to direct the entire machinery to then cut and then modify the DNA of a living cell or organism. So you can apply RNAs as well in the genome editing world.
Yeah, that's really, really interesting. I keep asking all these questions and I forget we've only been like a few seconds into this tour, so we better keep going. We still haven't seen any of Yunan's lab yet.
All right, so so this is here you can see behind those screens, that's the actual lab. And we are now entering the lab. Of course, PPE is necessary. You need to wear safety glasses, lab coats. Your shoes need to be covered, so then you are protected, and everybody's protected. So safety first. So we can go in. We can go inside the lab. Oh, and this is Yunan lab.
Ah, well, there you go. Well, we might get Yunan up on the screen so she can talk about her lab as well, and you can both help each other out.
Oh my
Okay,
gosh, so
hello.
great.
Good to see you again, Yunan.
Yes, so Paolo talked a lot about the molecular stuff that happen in a small volume of clear liquid, but our work is quite visual. So our end goal is to image mouse oocytes or embryos in action. And so we really need to keep them really happy and warm and acting as if they are still inside the body, even though the first thing we're going to do is to actually dissect them outside the sex organs. Yeah.
Fair enough.
So,
In the name of science, I guess. Just really quickly,
Yeah.
before I get you to keep on going, what's this? Why are we looking so intensely at this thing?
yeah, so this is a thermal plate that would, it's basically something that we put the dish on top of, so we can keep our oocytes and embryos warm. And then this one is a culture medium that contains the nutrients and pH buffer, everything our cells will need to grow happily. And yeah, so there's a dish with medium inside.
So that's just medium. It's not the cells?
Yeah, it's medium, and then the cells are inside, but the cells are so tiny that you can't see them
Okay, cool.
without a microscope. Yeah.
So what are these called?
So these are called electronic pipettes, which you see a lot in every wet lab, but we don't use them to transfer oocytes and embryos. We actually use this very historical technique called mouth pipetting. So the first step is we pour a very thin glass pipette that its diameter is just big enough to allow one oocyte to pass through at one time. And then we connect that to some filters and the mouth aspiration tube. So with this setup, we can transfer oocytes really easily, very controlled manner. Because if you use an electronic pipette, even the smallest tip will still be quite big.
Ah, and so if it's so small, what's to stop you accidentally ingesting some of it?
Yeah, good question. I had the same concern as well, but you can see we're connecting the glass pipette to a P1000 filtered pipette. So there's a filter in there, and we put a bit of extra tissue to stop things transferring, and obviously we'll change the filters quite often. So this is all approved by the health and safety, and it's totally legitimate.
I'm glad we're showcasing legitimate things here. Oh, where are we at now?
Yeah, so even though we keep the oocyte on a heat block and we try to protect them from light and with medium, it's still not the best just sitting outside. So we move them into these special incubators as much as possible because they have special atmospheric control of CO2 content and temperature, so they can grow really well. So for example, if you look inside an ovary at one time, you can find oocytes at different developmental stages.
Is that what we're looking at right now? Are these ovaries?
No, these are, if you go back to those follicles, so these are very immature oocytes enclosed with surrounding cells, so that's called a follicle. And inside ovaries, you find like these early stage follicles first, and then we take them outside the take them outside the ovaries and then culture them in the medium for 2 weeks, and then we get the next look, which is quite different.
And so this is where you're getting that pipette out to be able to look at like individual ones. Is that correct?
Yeah. So this is after 2 weeks of culture. You can see the surrounding cells have massively proliferated, and the oocyte is in the middle there, tucked nicely. It's trying to grow in size to prepare for fertilization. It really needs to grow in size for like 100 times of its original size. And that is a fluid-filled cavity called the antrum. So with the formation of antrum, it's signaling the end of the first development stage. And then here, we can use a really thin pipette to denude the oocyte. So
Sorry,
because
hang on. Did you just say denude? What does this
Yeah,
mean?
so-
As in like you make it
so-
naked? Or
Yeah, exactly like that. So if you remember from the last slide, the oocyte was barely visible because it was covered in a lot of surrounding cells, right? So we don't want the oocyte to be covered. We want to strip them naked by using a really tiny, thin pipette and going with the mouth aspiration, just lift it up and blow it out, and then we get rid of the surrounding cells. So these look really pretty and big because they're fully grown, but they are still immature. So after a night of maturation in the incubator, they would undergo a round of meiosis where they get rid of half of its genetic content into a really small polar body to create mature oocytes.
So meiosis, that's that process of reproduction, isn't it? Where cells start splitting to make more. Is that right?
That's mitosis, but meiosis is quite similar. But mitosis is to replicate. Meiosis is to get rid of half of the genetic content. And the reason why the polar body is really small, because now you mentioned mitosis is mostly symmetrical, right? One splits into two identical sized cells. But here you can see it's very different. The polar body is massively smaller than the oocyte, and that's because the oocyte contains almost all the nutrients and proteins that are necessary to sustain embryo development. And so they don't want to just get rid of all this cytoplasmic juice while extruding excess DNA. So it's a very smart mechanism to get rid of excess DNA without losing too much of the good stuff.
So at this stage, would you count this as one sample, or is this multiple samples? Are you going to just use one of these circles? Oocytes, sorry?
Yeah, right. That's a good question. And so these are individual oocytes that we grown from, harvested and then grown from one mouse. And we can do that with hormonal injection to to induce growth of multiple follicles. And that's the benefit of using a mouse as an organism. So from each ovary, we can get a lot of oocytes to work with.
Awesome. All right, let's keep going. See what else fun things we can see. What's
the exciting
here?
part of that's IVF, in vitro fertilization. So the sperm is swimming and trying to find the egg, and some of them are attached onto the egg and trying to get in.
So is this how it would work in terms of if a person would like to fall pregnant and they go with in vitro? Is this what they're pretty much going to be happening in the back end?
Yeah, so that's pretty much the first thing that they're going to try, to get a bunch of sperm and let them decide which one wins the race and get into the egg. But if this doesn't work, then we would move on to the next machine in the next episode.
Stage 2.
Yeah. Yeah, so this is the micro injection station, and what I think is the most amazing and important piece of machine in fertility. So basically we use this to inject stuff into eggs. So in a clinical setting, we would use this to inject a single sperm directly into the egg. So this really just facilitate things. You can't really get any easier with fertilization, directly injecting a sperm into an egg. The whole system is controlled by air pressure. So this is the pressure controller here. And you have two sides. So on the left side, I have the holding pipette that will stabilize the oocytes. So you see that long thing sticking into the dish, that's the holding pipette, and it's connected to air suction. It will help stabilize the oocyte. And then on the right side, I will have an even skinnier needle to inject into the oocyte. So on a daily basis, what I inject into oocytes are mRNA that encode fluorescent proteins tagged to structures of interest such as chromosomes. And so the purpose is to be able to visualize chromosome movement later on with the microscope. And so you can see here is the micromanipulator or the joystick or where the fun is. And that's a video
Whoa.
in action.
Is that how fast you work? Is this real time?
Yeah. You have to be very fast because you don't want to subject the oocyte to too much trauma that they die. And so this is really difficult. You have to control the air suction, control the needle size, and your hand coordination, and everything needs to be right for the oocytes to be able to grow normally after this.
It really is like a joystick game. So, okay, just really quickly, with that suction, can you accidentally suck up a oocyte?
Right, you can see a little bit of the cell going inside the holding pipette, but to be able to actually burst it, that would need a whole lot more suction. So that hasn't happened, although it might. Just with anything in science, this is practice makes perfect, and hopefully you don't kill too many oocytes.
So where are we now?
Yeah, so this is the needle puller. So I was just gonna show you how we make that really fine needle. So with the handling pipette, we pull that with the flame and then with our hand going apart. This one is a lot more controlled. So you see the metal, a heat filament that's heating up right now. So that's melting the glass capillary. And then the two carriages would move rapidly apart. I think it already did. Did you hear a loud bang?
Yeah, that was a little bit loud.
It was loud an hour ago.
Yeah.
So that was.
I'm so sorry, everyone at home. I hope that didn't wake up any small children or small pets. So okay, sorry. So the bang went off because what happened?
Yeah, so the top and bottom carriages moved rapidly away to pull the needle into two parts. Right? Does that make sense? So firstly, the tube needs to be heated, so it's more flexible, and then the top and bottom parts separate rapidly, creating that loud bang, and we have the fine needle in the middle.
Wow. So back in the day, are you saying that we used to, you could do this by hand by putting it over the flame? It's just this is much more accurate, or?
Oh no, we still use the flame to create the holding pipette. That one that I attach to the tube and put into my mouth. That one I create by the alcohol flame.
So this whole process of doing the eggs and that fast process you've just done and doing all of these needles and stuff, Alison would like to know, how do you train for this? Do you just get out a whole heap of eggs and just flow through it, or what do you do?
I'm glad you asked, Alison. This is a long journey. In the beginning, I had so much trouble. I had motion sickness looking under the microscope trying to identify the oocytes, because you find oocytes at all different stages and you just get motion sickness and eye problems. But now it's better, trust me. And with the micro injection, it works like a game, right? And I suffered because I didn't play many games growing up. So my partner bought me a Switch PlayStation, and we played Mario Kart and stuff to train for this. So now it's all working. It's just you have to practice.
Okay, so any of you gamers at home, I think this is a field you might want to think about getting into, IVF. Okay. Wow, that makes sense now that you think about it. And so you just use real oocytes that you work with, right? And you just keep on testing it and testing it. Whoa.
Yeah, it's really fun.
And so this is just you saving them for later, isn't it?
Yeah.
Cool. So where?
And now
Sorry,
we
go.
move into the microscopy room, which is where the magic happens. The clear liquid that we injected into the egg can now be visualized. And this room is usually dark to not interfere with imaging. But just so you can see, I've turned on the light. The first thing you can see is this massive box enclosing the microscope. So this is to keep the temperature stable inside at 37 degree, and it's custom designed for long-term live imaging. And oh yeah, no foam because you might get stripes on the images. There's some signal interference thing.
But we're not
Yeah, and
filming.
if
I
you
mean,
tap,
we're not trying to capture
yeah.
an image right now, so we're okay.
Yeah, so that's another chamber that's directly enclosing the dish of oocytes, and that's also connected to CO2 gas and moisturized air and temperature. So that little chamber is really well controlled. So I can grow our embryos a couple of days in there, and they can develop just fine.
And so you just film them? Is that kind of the idea with this, or just pictures?
Yeah, that's exactly the idea. We do live imaging. And so this is the control center where we can provide the light sources. So remember, there are fluorescent proteins in our sample introduced from the microinjection. And the fluorescent proteins can be red or green or whatever color, and they have properties that they can be excited at specific wavelengths, and then they emit light at certain wavelengths, and we can tell the program to detect such lights. So here I am imaging the first embryonic division of a fertilized egg, and I'm scrolling up and down because I want to show you it's a 3D volume. I'm capturing images at multiple planes and at different time points. So in the end, I get a really cool time-lapse video like this.
That is so cool.
Yeah, and the red was labeling the chromosomes, and the green was labeling the microtubules, which make up a spindle that physically pull the chromosomes apart into two sections of the cell. So that one was a symmetric division because that was the embryonic division, and this is oocyte meiosis. So this looks a bit different to just then, but the idea is the same. You have a green spindle that will pull apart the red chromosomes, and in this case, the spindle is positioned far off to the side. That's why the division is very asymmetric.
So interesting. It's such nice colors. Ooh. So is this?
This is.
Sorry, go for it.
That was a piece of art that I made for fun.
Oh, okay.
Just the
So,
perk of
so
working
does this
with.
show the different stages, or?
Yeah, so this is taken from a time-lapse images. So in the middle there wasn't any green because the spindle hasn't been assembled. So as the oocyte enters meiosis, assembles a spindle. So you can see that. And then the spindle elongates and the red chromosomes are moved apart. That's the process.
Wow. And so this process, Steph wants to know from the audience, how long does this take?
Right. So another perk of working with time-lapse live imaging experiments is that you can set it up during the day and then go home. So it will run overnight. So this process ran when we are sleeping. So it takes about 10 to 15 hours, and we do that at nighttime. It's really amazing, the feeling you go to bed thinking, what am I gonna find tomorrow? And then you come back to the lab and see really beautiful images.
Oh, that's really, really good. I guess it might not work all the time, but when it does, it's probably super exciting.
Yeah, you just forget about the times that they don't work.
Now, this might be a touchy topic, and I don't want to hold you on it too long because I know we've got so much to go, but how many times, like as a percentage, how often does it work?
Well, so I've been doing PhD for just almost a year. I would say the first half of the year, things didn't really work, but now it's pretty consistent that you've set it up. You kind of you learn every possible way of failing, and now you finally get some results. That's how it works.
Noted. So fail a lot and we will succeed in the end. And I guess that's also innovation
Yeah.
in itself as well. Alrighty, let's keep going. Let's see what else we're looking at. I think we were about to see somewhere else. Is this, Pilo, is this your lab?
Yes, that is my group. So I'm going to walk you through like a hypothetical experiment. This is a PCR machine. This is a very fundamental piece of equipment in every lab. And if you work in a molecular biology lab, you probably use this every single day. So this machine, what does, amplifies the DNA. So you set up a reaction and you basically amplify the DNA. And here you may want to image the product of your PCR reaction. And you see there all these white bands. These are pieces of DNA that we have amplified and we have separated, running them in a gel under an electric field. And so these little bands that you see, we moved on. But basically, those little bands that you see, here we go. So these bands are pieces of DNA of different sizes. The lower the band is towards the bottom of that screen, the smaller is the piece of DNA. And sometimes, like, this is an important information for downstream processing. You may want to purify that particular band and put it into a vector and so on. So that's why we need to visualize it.
It's really cool.
And of course, like, we print it because we want to keep a record of what we have done. This is gonna end into our lab book. And that's actually very, very important. Keeping a lab book is really important. This is a typical bench with a bunch of reagents, pipetters, timer, heating block, and that's the famous lab book.
I can see some photos in there.
Yeah. And these are like bacterial cultures. We use bacteria to amplify the vectors that we saw before, those circular pieces of DNA. We put them into bacteria and let them grow. In this incubator, this is set at 37 degrees. You can see this liquid is not clear, as it's a bit cloudy. That means that there are bacteria growing in there. And bacteria can also grow on a solid surface. This is a plate, and those little dots that you see there, each dot is actually a colony of bacteria, and they're clones. So basically, each colony derives from one single bacterium. They just amplified, multiplied overnight, and now created, its little family of clones. Yeah. Another piece of equipment that we have in the lab are centrifuges. We use these to separate, for example, bacteria or cells from the medium where they grow. We can use centrifuges also to separate DNAs, RNAs from the rest of the solution.
Sorry.
Yes.
What is Mumble 2? Is that just a fun name, or?
That's the name. That's the name of this freezer. So this is -80 freezer, which was baptized as Mumble 2.
Okay.
Don't ask me why. This was before I joined, so I'm not really sure. But yeah, so this is the name of this particular fridge, or freezer rather.
Oh, there you go. Fair enough. Okay, we'll keep on looking and seeing what's going on.
Yeah, so this is a low temperature freezer, -80 Celsius, and we keep tissue samples in here. We store DNA, RNAs. Now we're going to open one of these boxes, and we actually keep at -80 also the same cells or bacteria that we saw before growing. So we could store them in these little vials, and we can preserve them for a very long time if we keep them frozen. And here are just a bunch of racks and boxes. Again, these contain like a lot of tissue samples, most of them from mouse models that we generated, but also isolated RNA or DNA. It's all kept at -80, so it's well preserved for many years.
So what do you think's the most oldest sample you've got in there?
Oh, well, I'm really new here. So I joined CCR just a year ago. Some of the samples, I brought them from a previous lab, but I actually came to Australia 4 or 5 years ago. So yeah, I think that the oldest sample that we have here is probably five years old. But in my previous lab, when I was in the States, yeah, I had like samples that were like 10 years old or something like that. So yeah, very old samples.
There you go. All right,
Yeah.
let's keep going. What else we can see?
This is the histology room. So this is a fun room to be in because this is where we do our tissue processing. So once we collect tissues from a mouse, for example, we want to preserve it and we want to be able to section it and study it. So one of the first things that we do, we put it in this tissue processor. The tissue is dehydrated and is embedded in this station that we're seeing right here into a wax substance. It's called paraffin, but it's wax. So once the tissue is embedded into the wax, we can use these little guys here, which are called microtomes, to produce sections of those. So we can actually cut the tissue into thin slices. Wow. And this is the regular microtome, and this is a cryo microtome. So in this case, tissues are preserved by freezing them. They're not embedded into paraffin, and they're cut under, I think, -20 or more degrees Celsius. So they're frozen, they're cut frozen.
And do you keep it frozen or do you let it thaw out afterwards?
Exactly. So once they are cut, then you can process them at room temperature. If you want to do staining or immunostaining using fluorescent antibodies like we saw before, but throughout the entire processing until up to the point where you actually produce the final thin sections, the tissue needs to remain frozen. Yeah.
Oh, okay. Huh, cool.
This is another centrifuge. It's much bigger than the previous one.
Did it take more samples or bigger samples?
Sorry, can you say that again?
The centrifuge, does it take bigger samples or more samples? Why is it bigger?
It's bigger because it runs faster, so it spins faster. But also you can put bigger vials in there, but it's mostly because of the speed. So these are like high-speed centrifuges that can reach hundreds of thousands of G-force. So they're very sophisticated centrifuges. And the next one is a autostainer. So we do our most of our histologic staining, which is an automated process. You can see all these little containers. They, if you can go back, they contain several reagents, mostly are alcohol at different concentrations, and then the actual dyes. And that's how you do a normal staining of your sections to look at the morphology of the tissue that you're studying, that you're looking for.
Very interesting.
And you could do this manually. So I used to do this manually, and it doesn't take long, but it's tedious. But now that we have this machine, we can do this completely automated. So that's good. And this is actually a piece of the mouse, of a mouse embryo, and we can see that this is the urogenital system of a mouse embryo. So what we are seeing here are the dashed line, that's the ovary, so that's the gonad of the mouse. And in blue, that's the developing Mullerian duct. So this is the developing tissue that will become the uterus, basically. And in the mouse, you can see that this process starts at 11.5 days after fertilization, where like a few cells that are blue starts to appear, and then they multiply and travel down. And so the duct, so this tube elongates and migrates down until 2 days later, at 13.5 days after fertilization, the two tubes on either side of the mouse body, they meet in that region where later on the vagina will form. So the process is very fast. In the mouse, it's just a couple of days. But also in human, it's very fast. It's just probably a couple of weeks, 2-3 weeks. So it's a very, very regulated process that we still don't know much about. So what we are trying to do now is trying to understand how this process occurs. And here on the left, you have a section of the same tissue that we just saw. In blue, that's the Mullerian duct, so the future uterus. But next to it, you can see the Wolffian ducts. This is the tissue that I talked earlier. This is the tissue that in males will form the male reproductive tract. So again, in the embryo, we have both, and then one or the other will continue growing and developing depending on the sex. And this is an electroporator. So this is what we use to perform our CRISPR or genome editing experiments. So this machine is what it does. It applies an electric pulse to what we put in between these two electrodes. So we have the positive and negative electrodes. In between, we put cells or embryos, and that we can see here actually. So this is a close up of these are zygotes. So these are single cell, one cell embryos. This is the oocyte just after the fertilization by the sperm. So we put these zygotes. If you can go back a little bit, we put those zygotes in between those electrodes. And when you see the bubbling occurring, that is when we apply the electric pulse. The electric pulse, what it does, it permeabilizes these cells. So temporary pores will appear in the cell surface, allowing the CRISPR component that are in this liquid to penetrate and go inside the cell. And the CRISPR component at that point will do their job to edit, to modify the genome of those cells. So this is how we actually generate a genetically modified mouse. After this step, we will culture these embryos and we'll transfer them into a recipient female that will deliver genetically modified pups later on. And before there, you saw the cells that on the left, this is the after electroporation. These are the zygotes. The green ones are those that have received the CRISPR components. We use, in this case, fluorescent components to be able to monitor the efficiency of the process. And the right picture is to show you that electroporation is not the only technique that we use to make the CRISPR component get inside the cells. We can also use the micro manipulator that Yunnan showed earlier. So we actually mechanically and physically inject these components inside the embryos.
Now, between those two techniques, which one's more efficient is probably the word I'm going to go with.
This is a very good question. So first of all, we use the electroporator when we want to insert small edits, some small pieces of DNA that would go inside the zygote. And this is because the pores that actually open up after the electric pulse, they're quite small, and so only small molecules can get inside. If we have like a big molecule of DNA that we want to insert into the zygote, then we probably would go with the microinjection technique. So we actually inject them using the injecting pipette. In terms of efficiency, it really depends on the type of experiment that you're doing. Electroporation is really efficient, very, very efficient. So probably is more efficient than the microinjection. But also you need to, but also the type of modification that we're doing using the electroporator is, let's say, easier. So it's smaller, so it's easier compared to the type of modification that we're doing when we use the micromanipulator. So it really depends. It varies a lot. Also, it varies depending on the target that you want to use, on the gene that you want to modify. There are a lot of variables at play here.
So it sounds like both mechanisms are both just as useful as each other, and they both serve both pros and cons depending on what you're getting up to. And
yes.
Fair
and
enough.
there is a lot of thought into what kind of technique do I use, what kind of modification do I want to perform. So there is a lot of thinking before doing the actual experiment, a lot of design.
So cool to hear. Now, I am aware of time, and I'm cutting it close here, so let's get back to the video so I can ask you some questions.
Yes, so we'll go fast. So then this is a cell culture room where we do our in vitro work. So we culture cells in plates that we'll see. These are just the regular equipment. This is the incubator. We keep the temperature at 37. The CO2 percentage is 5%. So this creates a nice environment for the cells to grow. And in here, you will see the cells. They are kept in flasks or in plates. And so the environment is controlled. And these are the cells that are either, these particular ones, I believe, are from actual human samples, I believe. But we can keep, any type of cells in that incubator. This is a biocabinet where we actually do our cell work. We don't, open those flasks in the air. We put them inside these cabinets to prevent possible contaminations from outside organisms, and so to keep everything clean and sterile, we go inside those hoods. This is a cell counter, which will count the cells automatically. And this is a confocal microscope, much like what Yunan showed earlier. This is a bit of a different setup, but it basically does the same things. And these are other microscopes. These are fluorescent microscopes. You can see that they are quite smaller than the confocal, but they do pretty much the same job, just at lower resolution. And at the end of the day, we wash our hands and we're ready to exit the lab. We put our dirty towels and lab coats where they need to go, and then we head out of the lab.
Now, a very controversial topic that I've heard a lot of my science friends been talking about is what happens to your lab coats? Do you have someone that cleans them for you, or do you have to clean them? What's the deal?
The lab coats will go down to the cleaning station, and yeah, there are staff people that will take care of that. So they will be autoclaved, washed, so they will be sterilized, and so they will come back to us clean and ready to go again.
Wow, that's pretty cool. You have someone doing the washing for you. That's great. Now, as we come to the close of our session, I do want to ask you folks one big question each, just questions that have come out from the audience. Firstly, maybe Yunnan might be able to answer this question for me, but it might have to be Pillow. I'll let you folks decide who this goes to. But Tom from the audience wants to know, your ultra low freezer that you were talking about earlier, is that the same temperature that people use for vaccines or holding vaccines? Do you folks know? I know that's a bit outside of your fields.
I guess it depends on what the vaccine is made of. If it's with the mRNA, I think with the Pfizer one, it's mRNA, and you have to keep that at ultra low temperature to keep it stable. And with other stuff, it might be require a higher temperature. I hope I'm not wrong, Pillow.
No, you're exactly right. So it depends on what the composition of the vaccine is. If the vaccine is RNA based, then yes, that's exactly the freezer that you need to use to keep that that vaccine at.
Noted. And keeping with the vaccine theme, it seems, there is one other question asked from Ali, who says, isn't the PCR thing they use to test for COVID? Is that the same machine? Is that still using the same technology?
Yes, exactly the same. So the PCR is really the base procedure that is done used in any lab. Every time you need to amplify a piece of DNA, that's what you use. And so it's very common, and it's very, very, very useful, of course.
Thank you. And I am going to ask one more question. I know that we said last question, and I always get in trouble from the team when I say one last question. Final question to you both, and I'm going to start with Pilo for the answer on this one. My question is, of the lab, what is your favorite technique to play with or work on?
So I'm an old school guy. What fascinates me a lot is to see how cells develop into tissues. So I think that the thing that amazed me the most is just actually visualizing tissues under the microscope. So basically the confocal, when I see images under the confocal, or just under the normal microscope, I think that that's the most amazing thing, to see how cells develop and organize. So there are a lot of very cool techniques, genome sequencing, and whole genome sequencing and transcriptomics and everything, bioinformatic techniques and everything. But actually seeing something, I think it's the most satisfactory thing in the lab, personally.
And what about you, Yunnan?
I can totally agree. I guess the first thing that attracted me to developmental biology is also just thinking, how can one cell become 37 trillion cells that make up of us, and all the tissues, organs, they are all different. And especially when you stain tissues with immunofluorescence, you can see all these cells light up in different colors. That's just amazing. And I mean, the microinjection rig is really cool, but it's a love and hate relationship because it can be too difficult sometimes. But the confocal, it shows you really nice images, so I love that.
And I guess there are a lot of confocal microscope competitions out there with people showcasing all of their photos. So I'm sure you're not alone in sharing the love of those photographs that come out and just all the beautiful pictures. And a lot of the individuals, a lot of us out there in the world are visual people. And so seeing it physically, something we can't usually see, I can totally relate as well. Now, unfortunately, that does bring us to the end of the tour today. I am already running overtime, typical me, but I want to make sure that we have an opportunity to talk to you both about if, we mentioned at the beginning, Yunnan, that you yourself found out and wanted to join this lab after a tour. So if people want to find more about you both, where might they go, Yunnan?
So the best place would be our website. If you type in UQCCR fertility theme, you'll be able to find both Pello and our lab, led by Professor Hayden Homer there. And myself, I am on Twitter. So if you want a chat, I'll be there as well.
Awesome. And Pilo, anything else you want to add to that? Or did she tick it all boxes?
Yeah, she did. She did. So I don't have a Twitter, so I'm a dinosaur here. But yeah, so the website is the best place to go.
Excellent. Well, thank you so much, both of you, for allowing us into your labs. And I hope that you disinfected all of the camera gear on the way out. Don't want you folks getting in trouble or having random eggs floating about. That's probably not the right words to have used. In any case, we're going to move on. Thank you so much for your time. Now, audience, thank you for hanging around with us tonight on what is the last Pint of Science live stream for 2021. I hope you've really been enjoying yourself and checking out all of the fun things that we've been having on. If you haven't had the chance to do so, don't forget to head to our YouTube channel or head to our website. While you're looking for our YouTube channel, or you might not be ready to have some more science in your face, you could probably subscribe or maybe like, just some fun things you might want to get up to. But we actually don't finish the festival in terms of our pre-recorded content. There's still a few more things coming out this weekend. So don't forget to have a look out for Podcast and Mug of Science. Both of these will be on our social media, so have a look out there. Or if you're already subscribing to our channel and getting notifications, you'll get told about the Mug of Science video that's coming out tomorrow. There's also a special one with the two co-CEOs, so myself and Tom. So have a look out for that as well. Now, as a registered not-for-profit powered by volunteers, we at Pint of Science rely on the community to keep the science going. I mentioned this earlier, but a donation will really help us enable the future of Pint of Science and keep these types of events going. And hopefully once we get back to a more the new normal, I guess, of being able to go out to the pubs and hang out together as a group, your donation is really going to help us get that next level and be able to come back into the pubs. If you're more of a merch person, then I strongly recommend checking out our merch that also supports us. We have shirts, we have pint glasses, we have pins. So please check it out, and it includes all of this year's artwork, and there's also some previous year's artwork as well. So have a look. Now, once again, don't forget to check out our social media pages in case there's anything of interest or you want to keep in contact with us and find out about any more Pint of Science content that comes available. That does bring us to the end of the show. Thank you for sticking around with us. I hope you've had a wonderful Pint of Science festival. But that is all from me today. Thank you so much, and we will see you on the social medias. Bye, everyone.